So good morning again, and welcome to today's Next Talk Bioinformatics webinar, Bacterial Nanopore Sequencing Data Analysis. Thank you all for joining us. I'm Mingqi, and I'm the moderator for today. To make sure everyone has a great experience, here are a few things to know. This session will run for about 40 to 50 minutes, and we will have a Q&A session towards the end. If you have any questions during the webinar, please feel free to type them in the chat box, and we will address them after the presentations. Lastly, please keep your microphone muted throughout the session. In today's webinar, we are happy to have Ms. Jiaxun as our speaker. Jiaxun is the Bioinformatics Manager at NextGene Scientific, and she has over a decade of experience in bioinformatics field. Today, she's going to walk us through how we can analyze bacterial nanopore sequencing data using the bioinformatics tool, Epi2Me. Without further ado, let's start the webinar today. Jiaxun, the floor is yours now.
Hello. Is my voice coming through okay?
Yes, everything is okay.
Okay, so let me share my slides. So I think my slide should be visible to everyone here. Okay, so I think we have rather small groups today. So please feel free to let me know if, let's say, there's any hiccups or any questions that you have. You can also put the questions into the chat box anytime. We will come to the questions Q&A at the end of the session. Okay, so for today, we are going to talk about a little bit on the nanopore sequencing. We will be spending a few minutes to go through the nanopore sequencing and the technologies. So this nanopore sequencing and also the technologies, we have also gone through several rounds in our past webinars. For participants who have just joined our session today, we will have a very brief rehash on the nanopore sequencing and technologies. And then we will be more focusing on the microbial isolate sequencing solutions, the NOMIS protocol. And we will be into the data analysis by using the Epi2Me software at the end of the session. So without further ado, let me introduce you Oxford Nanopore Technologies. So Oxford Nanopore Technologies, or in short, the ONT, is the UK-based company. And then the core technologies from ONT will be the nanopore sequencing products. So the DNA sequencing technology is rapidly gaining traction since the development of it. And this actually reflected in more than 14,000 of scientific publications, and then the abductions in more than 125 countries as of the end of 2024. So continuously, ONT invests in more and more technologies, developing more technologies, including the proteomics and metabolomics. So the users of ONT actually have used the platforms to understand the biology in many different fields or domains. So that include the pathogen research, the epidemiology, and then studying the environment, like the samples from different regions, including the space, and also different industries. Players from different industries has used the platforms for different purposes. So the core of these technologies, why many users can use the same technology for different purpose, basically three main different benefits. So compared to other DNA sequencing technologies, so Oxford Nanopore offers richer insights. By sequencing the same samples, at the same time, we can get the information regarding the DNA sequence, and then at the same time, we can also understand the epigenetics of it to see whether any of the modifications or methylations have been there in the DNA or RNA molecules. And then it's also faster time to results. So compared to other DNA sequencing technologies, ONT provide real-time sequencing. So that means while the sequencing is still ongoing, we already can get the data directly from the sequencing run, and then further look into the analysis of it to get the information through data analysis without requiring us to wait until the full sequencing is done. And then it also very accessible and very affordable. So the devices from ONT are basically among the smallest in size. So it's very easy to set up in any labs or even in the office. And also it's also very easy to kind of, if let's say you want to set up a lab and then after that you want to relocate it to somewhere else or even bring it to field to do the sequencing, I think is quite accessible. So how the nanopore sequencing work? So here's the heart of the technology. So as Nim mentioned, the technology is named after this protein called the nanopores. So the pore, basically the pore proteins has a channels inside the protein, so that allow the DNA molecule to pass through the membrane, pass through the proteins. So these proteins are fixed on membranes. So from one side, the DNA molecule will pass through the channels to the other side. And then while this happens, we can measure the changes in the electricities within the channels. And then that basically is our data that is used for base calling. So how it exactly it works. So I have some animations over here. So the nanopore proteins is fixed on membranes, supporting membranes. And then our samples or DNA or RNA, it will bring over to the nanopores by using a motor enzymes, and then the enzymes will also unzip or unwind the DNA molecules or RNA molecules, and then let the strain of the DNA or RNA pass through the channels. And then while the molecules are passing through the channels, the electric signal will be turned into base call through the kind of, we use computer and software to control or to collect the data at the same time. So this actually bring the benefits of direct sequencing of the DNA or even the RNA molecule itself. And then without any PCR or without any amplifications, we can directly measure the orders of the DNA or RNA molecules in terms of their sequence. And then at the same time, we can also measure whether any of the bases are methylated. So to look into the epigenetics of the samples. So, and then this form or this way of working on the DNA sequencing, it also read length agnostic. So that means even though it's called as the long read sequencing, but actually the technology can sequence any size of DNA fragments. So it can sequence as small as less than 100 base pair. It can also sequence all the way to few KB or MB in size for the DNA samples. And then because the sequencing, it happens real time, so we get the results when the sequencing is still ongoing. So that means we can also tell whether the DNA molecule that we are sequencing are the right one that we want to get from our samples. So that develop a targeted sequencing approach called the adaptive sampling. So this adaptive sampling is a way to enrich or deplete based on our regions of interest. So in a samples that might be kind of the full genome of an organism, but we might not need to sequence everything to get the information that we want. So how to target certain regions that we are interested in. So oftenly we use maybe primers or probes to capture the target regions, or sometimes we might even use technology like CRISPR to cut out the target regions. But those methods normally will be disrupting the native form of the DNA or RNA molecules. So with this adaptive sampling, we are using the whole DNA samples, and then we inform the software that what are our target regions. So the software will control whether a nanopore is sequencing the molecules that we want or the regions that we want. If let's say it's not the region of interest, the software will send the signals to the nanopore so that the DNA molecules from the pores will be ejected or will be rejected from the sequencing process. So if let's say the DNA molecule that passing through the nanopore is the right one that we want to get, then the sequencing will be continued. So this is one of the very unique way to do the targeted sequencing by using the nanopore sequencing. So all this actually that happens inside the flow cell. So all the nanopores are fixed into the flow cell. So there are two types of flow cells from ONT. So one is called the mean ion flow cell, another one is called the primary ion flow cell. So the difference between these two are basically the throughput, data throughput. So the mean ion flow cell normally can give us roughly 10 to 20 GB of data. And then primary ion flow cell will be giving more than that, reaching 100 GB of data. So mean ion normally is suitable for small genomes, especially like our topic today, the bacteria or microbial genome. And then the primary ion flow cell can sequence a very large genome like human genome and etc. So depending on the requirements, so we have the options to choose mean ion flow cells or primary ion flow cells. And then based on the flow cell chosen, that decide what devices that can be used to do the sequencing. So we load our samples into the flow cells, and then the flow cell will be put inside the sequencing device. So there are multiple sequencing devices available from ONT. So from the very small scale, the mean ion MK1D. So this is a palm-sized sequencing device, and then it can use to handle one mean ion flow cell at one time. So we also have another larger throughput of mean ion sequencing device. It's called a grid ion. This grid ion comes with the compute power. And then for primary ion flow cell, we have also multiple options in terms of the sequencing device. So the primary ion flow cell, primary ion 2 solo and integrated. So both these devices can be used to handle two primary ion flow cells at the same time. So it's like we can sequence and get around 200 GB of data at the same time. And then the primary ion 24 will be able to handle 24 flow cells at one run. So depending on the throughput requirements, different devices can be used or chosen from over here. So back to our topic today, microbial sequencing or bacteria. I think we will be more focusing on the bacteria for today. So just to rehash a little bit on our samples. So how we perform the whole genome sequencing or microbial genome sequencing. So we might have samples from different sources, like maybe clinical samples or sample from soil, sample from waters. Normally, these kind of samples will contain multiple organisms. So it have mix of maybe fungus and also bacteria inside, or even virus and maybe some other organism as well. So now this is our how our sample looks like in most of the cases. And then if let's say we extract the total DNA from the samples and then do some amplicon sequencing, this is one of the way of doing the metagenomics study. So we want to identify the members inside the samples. So this one, the amplicon sequencing, we normally will be using certain marker genes. And then from the marker genes, we will be able to tell what are the members inside the samples. And then at the same time, actually, we can also do the shotgun metagenomics. That means whatever that we have extracted from a samples, we will sequence everything. So from the sequencing, the data we can tell who are they inside the samples, and then what are the genes or what are the genomic content that are available inside the samples. So this is both of these are considered metagenomic samples or metagenomic studies. And then the more I think traditional way or conventional way of studying a mixed samples, normally we will be doing one round of isolation first. And then each of the pure isolates or pure samples, we can further do some DNA extraction, and then kind of one sample we will have the DNA content from one single organism. And then if we do also the amplicon sequencing, this is considered a targeted sequencing, and then normally the purpose of running the amplicon sequencing in this case is to identify who are or what is the species inside the samples. So it's for species identification. And if let's say we have the DNA samples from each single species or single isolate in one tube, and then we extract the total genomic DNA from the samples, and then we do sequencing. So this is the whole genome sequencing. So the data from whole genome sequencing supposedly all come from the one single species. And then after we obtain the data, we will be able to assemble the data back into the original form of the one single species. So today's we are more focusing on the whole genome sequencing for the microbes. So this is some of the possible way of recommended way to handle the microbial whole genome sequencing. So ONT has provided some guidelines on how to run the whole experiments end to end, starting from how to prepare the samples all the way to perform the sequencing and get the assembled genomes from the analysis. So this is the steps involved. So normally the first step is from the samples, we isolate the genomic DNA and then proceed to do the library preparation. And then once the preparation is done, we will load the sample into the flow cell and then put the flow cell into the device and start the sequencing. So when the sequencing is still ongoing or perhaps gather sufficient data, then we can start doing the data analysis. So this process, normally it could take a few days. So depends on what is the methods that is used to isolate the genomic DNA. It could be as fast as maybe 1 to 2 hours. And then that library preparation normally also within 1 hour to 2 hours. And then that means we can actually finish up from genome, kind of the DNA extraction all the way to prepare the libraries and load into the flow cell within half day. And then once the flow cell is loaded and then start the sequencing, then we can expect to see the data by the end of the day. And then to get sufficient data, normally we can let the sequencing keep on running for up to 72 hours. So how much data that we should get from a sequencing? So that would depend on how many samples that we load into the flow cell. So based on the recommendations, we should have at least 50x genome coverage per samples. So that means that's roughly 500 MB of data per bacterial genome. So assuming the bacteria is of normal size of 5 MB. And then the base calling should be in the high accuracy mode. So this 50x coverage should be sufficient for adult analysis using the epitome. So this include the assembly of the chromosome and plasmids, the AMR, the antimicrobial resistant genes identification, and then the core genome or whole genome MLST or SNP typing. So in terms of the samples or the genomic DNA extractions, there are multiple recommended methods or protocols shared by ONT. So depending on your samples or what is the target organisms that you want to sequence for the whole genome. So if let's say there are bacteria, normally we will be using the bacterial DNA extraction protocols. And then there are also bacteria that is very hard to lyse organisms. So there are also protocols available and also the protocols for yeast or fungus. So if you are interested, I think the protocols are shared by ONT freely to the public. So for ONT, they have also benchmarked on their protocols. So using 24 samples from Salmonella, Listeria, and etc. So it's a mixed kind of bacterial samples of different genome sizes. So they have obtained the samples from the sources here, listed here. And then they have done the DNA extractions by using the beating methods. And then they have kind of obtained the genomic DNA. So even though nanopore sequencing is not very demanding in terms of the samples required, still it's best to have a good DNA samples before we do the DNA sequencing, because that could guarantee the better results at the end. So this one, some of the samples or most of the samples are kind of managed to get back the good quantity and good quality DNA. And then some do have a slightly lower throughput, like the Xinjie. So they have run the sequencing by using both the primary ion flow cell and also the mean ion flow cells by using two different devices. So they have run it 3 times and then try to see whether the results are reproducible. So these are their benchmark results. And then they have also run the analysis by using the Epitome workflows or pipelines. So the pipelines basically able to identify the whole genome of it and also identify the plasmid sequence of it. So the more information can be also obtained from their resource portal. So the data analysis, so this is our focus for today. So from ONT to handle the Oxford Nanopore sequencing data, actually the ONT to prepare some solutions to do the data handling and also analysis. So for Nanopore sequencing users from ONT, we have 2 different software that can be used. So one is called the MinNo and another one is called the Epitome. So MinNo is the software that is used to control the sequencing device. So normally we will need to install this MinNo software when we run certain sequencing. And then for Epitome, this software is used normally for data analysis. So it comes with many different analysis pipelines. And then it can handle the nanopore sequencing data directly from the sequencing run. So both this MinNo and Epitome software come with graphical user interface, it's point and clicks. So you don't need to worry about any bioinformatics commands that you will need to execute during the process. So everything just point and click, very easy, user friendly. But for bioinformaticians, if let's say command line is still something preferred, both the pipelines and also the software developed by ONT, they are also come with the command line interface. So for users who prefer to run using the command line interface, is also still be able to run accordingly. So this Epitome software is free to download, it's open for the public, and then is downloaded as the standalone desktop application. So that means we can install it in our computer and then without any restrictions. And then it has pre-packaged with more than 15 different workflows or analysis pipelines. And then the setup is quite easy. So normally we just need to download the installer and then follow through the installation process accordingly. And then this software can be installed in many different operating systems, including the Windows, the Mac OS, and also the Linux. So this software actually can handle the analysis run locally in our computer. So it depends on where we install the software. Normally the analysis can be run using that particular computer. But in case that some computer might not have sufficient computational powers like the CPU and also the memory, then we can also run the analysis using the cloud provided by ONT, free. And then everything is also handled by this Epitome software. So we do not need to worry on how to set up the cloud instance or how to upload the data. So everything taken care by the Epitome software when we have it installed in our computer. So in terms of the analysis pipelines or workflows, those are the ones that already developed and then can be used directly. So these include the one that is used for human genetics, some basic tasks, targeted sequencing, transcriptome, and for our topics today, the bacterial genome or microbial genomes, we have one specific pipelines, which is the WF bacterial genomes that can be used to handle the microbial whole genome sequencing data to help us assemble the data into full genome and also to profiles or characterize the genomes accordingly. So for users who prefer command line, so here are some resources available also for you. So these pipelines are also available directly from their GitHub repositories. So normally we just use the Dockers or the Singularity containers to run the analysis. So we normally do not need to worry about how to set up the software or pipelines because everything has been contained in Docker containers and etc. So for analysis using the Epi2Me, so let me show you on the Nanopore sequencing data a little bit, and then how we can get the Epi2Me software, and then how we perform the whole genome sequencing analysis for the microbes. So for the data that I'm going to use for the demonstration soon is actually a demo data from the pipeline itself. So this data contains 2 samples. So it's one is using the barcode 01, another one is using the barcode 02. So inside the folders of barcode 01 and barcode 02, both of them we have one FASTQ file. So the FASTQ is basically the file format for most of the sequencing technologies. So we can put our folders in the arrangement like this one. So some backgrounds of these data sets. So this actually a very relative old data sets from ONT. So they have done kind of the data obtained is before 2022. So they have improved or modified a little bit throughout the time to feed it into the pipelines. So one thing to note that is the base core model for barcode 01 or sample 1 basically is added information. So it's not the native sequencing information. So because the sequencing data is quite old, so they fix it so that it can be analyzed directly by using the Epi2Me software. For the Nanopore sequencing data obtained recently or these few years, normally we won't have any problems to load into the pipelines to do the analysis accordingly. So the pipelines that will be used, including multiple steps, soon we'll be looking in more details into this one. Also want to highlight one thing is that currently the pipelines minimum requirements will be CPU, 8-core CPUs, and also memory of roughly 32 GB of RAM. So this one has been increased a little bit. So if you have seen the pipelines or the older version pipelines, the older versions of these workflows actually can cater only bacteria. So they have improved it so that the same pipeline can also be used to analyze the fungal genomes. So with the improvement, they also have set the minimum requirements from 16 GB to 32 GB. But for users who are analyzing bacterial genomes, even though the computer might not have sufficient 32 GB, most of the time, pipeline is still working for the bacterial data sets. Okay, so let me try to bring you to where we can get the Epi2Me software. So to get the Epi2Me software, as mentioned, is free for public to download and to use. So there's no limit who can access, who cannot access. So let me share the link into the chat box over here. So this is the consider the websites where we can download the Epi2Me software. So when we are here, we just need to scroll slightly down to the page. There's a download button over here. We just need to click the download button, and there are different installer for different operating system. So depending on what is the operating system of your computer. So if you are using Windows, then you should be downloading this particular installer. And then for Mac computer, depending on the chips that you have in your computer. So if you are having a relatively old computer which is still using the Intel chips, then this is the installer for the computer. And then for the Apple chips, M1 all the way to I think now it's M5. This is the installer for it. And then if let's say you are using Linux, so depending whether you are using Ubuntu or Debian or Red Hat or Rocky, so there are different installer for each of the Linux as well. And then how to do the installation, from the same page, you can see an installation guide over here. So we just need to go into this installation guide. So the process on how to set up the Epi2Me, including like what are the kind of the message that you will be received while installing the software, are all kind of documented over here, so in very detailed form. So just need to follow through the process. Just one thing to note that for the installation process, some cases or some computers might need to do computer restart to enable to finish up the installation process. So I would encourage everyone to install the software when nothing else is running in the computer. So if you are running certain things in the computer, I would suggest maybe you can put the installation aside first. So there are also different documents for installing installations for different operating system over here. So just need to follow through the process, and that should be done. So once it is done, the software should be opened up directly when you open up the software accordingly. So the interface is very easy. So let me enlarge it a little bit. So once you open up the software, normally the first page you encounter would be a login page. So basically we can register a free account just to log into the software, or else we can also use the guest account to log in. So once it's logged in, then we will come to this dashboard page or the home page. So where our status of the computers are listed over here, and also some activities that are ongoing. So if let's say there's any updates of the software that is available, it will also be shown over here as a banner. So we can proceed to update the software if we want. So, and then at the bottom of the homepage, we can also see some latest news from ONT. So what are the latest software available? And then any data sets that they share for users to try out also. So this is the dashboard, and then on the left side, the panels over here. So we can see one setting buttons at the bottom left. So this one actually helps us to manage the software on the computer resources allocated for this software. So we can see where the data analysis will be carried out when we run the data analysis. So normally is by default, it will be in our C drive under our user account. And then, of course, if you want to switch to a different working directory, you can also do it over here, set it accordingly. And then normally I will also take a look at the local tab over here, just to make sure that the system are set up accordingly. Because the software rely on two different dependencies. So one is the WSL, the Windows Subsystem Linux, and also the Nextflow job scheduler. So these two might go into dormant mode or sleep mode when it's not active for certain times. So sometimes we just need to come to this local tab in order to wake it up again if let's say it has gone into the sleep mode. So how to wake it up? Normally you just need to open the setup and then retry so that both the status change to ready, then the software is ready to be used to do analysis. We can also set the maximum computer resources that can be used by Epi2Me to do the data analysis, like how many cores we want to allocate for the analysis, and then how many total memories that we want to allocate for the analysis. So this one, normally I won't be maximize the whole system for Epi2Me to do the analysis. Normally I will leave some space or leave some memories so that the Windows can operate like usual. So this is the setting, and then actually we have the dashboard over here in the home button, through the home button, and actually we only have two buttons left, so very simple interface. The launch button and also the analysis button. So launch button is when we want to start certain analysis, and the analysis button is for us to view the results of the analysis that have been carried out. So the launch button over here, once we click on it, then we will be seeing the workflows of analysis pipelines that are available in Epi2Me. So this one, actually, many pipelines have been developed by ONT, and then they share directly in Epi2Me. So for some community users, they have also developed their own pipelines and then share it to other users or other collaborators through GitHub. In that case, users can also import the workflow into Epi2Me directly from GitHub. So for the workflows that is imported or developed by community, basically you might be seeing the tag of community over here. So these are the pipelines that are not directly developed by ONT. So the support of the pipeline or the community tools are all through the developers of the software accordingly. So the rest, each of these box represent one workflows. So you can see for each of these box, there are two icons at the lower right corners. So the computer icons means the pipelines has been installed accordingly and ready to be used in the computer. And then the cloud icon means the analysis can also be carried out by using the cloud resources provided by ONT. And then depending on what is the analysis that we want to carry out, we can select the pipelines from the catalogs over here. So for today, we are going to use the bacteria and fungal genome pipelines. Or you can see from the tag is the WF bacterial genomes. So once click into this, if let's say this is the, I think if let's say this is the first time you are using the Epi2Me and then the workflow is not ready in the Epi2Me, we can do the installation over here using the install button over here. So one is installed, we can see the full documentations of the pipelines over here in Epi2Me. So this include the brief introductions or the step-by-step on what this particular workflow is going to do for you when you execute it. So for this Epi2Me bacteria and fungal genomes pipelines, basically it helps to pre-process the nanopore sequencing data before it do the assembly and polishing. And then furthermore, it can help to do the characterizations and also the genome annotation. So it also have actually an alternative pipelines or workflows. It's called the reference assembly. So in case your samples, you are not going to assemble the data back into the full genome, you just want to see the difference between your samples and a reference genome, then we can also use the same pipeline for different mode. So it's called the reference assembly mode, where we can ask the software to map the data against a reference genome and then perform the variant calling by using the software used over here. And then in this documentation, we also have more details like what are the input examples and then what are the output files that we'll be getting from the analysis once it's done. And also the pipelines overview where it will help us understand what are the steps that will be carried out in more details, including the software and parameters used in each of the steps. So for bacterial genome or fungal genome, the first step, it will pre-process the data to make sure the data that is used for assembly are of good quality and then should be the good one for assembly. And then depending on the analysis mode that we select, so it can proceed to do the default de novo assembly, which involve the steps of assembly, contact with orientation, alignment, and also polishing. So optionally, we can also choose another analysis mode. It's called the variant calling mode or reference-based mode. So this one, the pipelines will help us align the data against the reference and then do the variant calling to identify whether there's any SNPs or insertion deletions and etc. So the same pipeline can also help us identify whether the samples carry any plasmids. And then if let's say yes, then it will also try to tell you which sequence belongs to the plasmids. And then the genome annotation, this one is used to identify the genes inside the genome. But this one, they have updated it to use the Bacta software. So based on my research, this software is used for bacteria or prokaryotic genome as annotation. So that means if you are having fungal genome as your samples, this annotation might not be very suitable for your case. So I would suggest after the pipelines is done, after you get the full genome for your samples, maybe another round of genome annotation might be required in addition to this one provided by using the Bacta. So at the same time, the pipelines will help to analyze the samples for isolate mode. So the isolate mode, that means the software will try to identify the MLST sequence typing of the samples. based on the PubMLST database. And also it helps to identify the species by using the sour mesh database or pipelines. It can also help to profile the AMR, the antimicrobial resistant genes or variants inside the samples. So from here, we can directly tell whether our samples is carrying certain antimicrobial features. And also, if let's say the samples has been identified as Salmonella, then a further serotyping will be carried out to identify the serotypes of the Salmonella samples. So additional information like the troubleshooting guide and also the frequently asked questions are also available in the documentations over here. So yeah, and over here to carry out the analysis, normally it's very easy. We need by default the analysis will be carried out in our computer. But if let's say we choose to run the analysis by using the cloud, then we just need to switch over to the cloud over here. Then the analysis, once we have set up, then it will be carried out in the cloud resource provided by ONT. So this one, I'm going to use my computer over here. And then before I start the analysis, just want to show you that there are some options to help us manage the pipelines or workflows over here. So we can run certain demo analysis just to try out the analysis pipelines and to check whether the pipeline has been set up accordingly in our computer. And we can also check whether the pipelines, whether there's any updated version of it, switch different version, and then delete the workflow if we are no longer requiring it in our computer. So this one can free up some storage space in our computer, and also some other options to help us manage the workflow accordingly. So this one, I think they have recently updated to version 2. So for users who have version 1 in the Epi2Me, I think some users have complained that the direct updates might not be so smooth over here. So in case you encounter any problems to update the pipelines to version 2, you can delete the workflows, the old workflows here, and then install the new workflow once the older one is already deleted. So that can help to fix some of the errors over here if let's say the direct updates is not available. So once the pipelines is ready to be used, we just need to click the launch button over here. And in this setup page, you can see most of the parameters have been pre-configured. So they have selected the best options for the normal use case for bacterial genome assembly. So normally we just need to select our input file. So our files basically should be inside the computer with the software. So we need to go to where we store our data. And as mentioned, we have the demo data from the same pipelines. So we have the folders of this isolate FASTQ. And then inside these folders, we have two samples, barcode 01 and barcode 02. So we just need to put the FASTQ file into each of the sample folder accordingly. Then we can select the whole folder as our input. Then the analysis pipeline will be carried out for all the samples inside the folder. So we do not need to run the analysis one by one, because most of the time we are able to multiplex maybe up to 24 samples in the mid ion flow cell or 96 samples in the primary ion flow cells. So if let's say we have 96 samples and then all requiring the data analysis, then these functions can help us kind of quickly select our whole folder of the sequencing data, and then the analysis will be carried out for all the samples inside. So we don't need to run the analysis pipeline one by one. So once this input file is selected, then basically it's ready to be run. So we just need to click the launch workflows over here. So, but before I start the workflows over here, just want to walk you through some of the parameters over here in the pipelines. So for some experiments, as you can see, our samples are all named according to barcode. So sample 1 is barcode 01, sample 2 is barcode 02. If let's say we want to name the samples accordingly, we can put in a sample sheet to tell the software barcode 01, and then we give a name to the samples, and then barcode 02, we give a name to the samples. So this can help us rename the data accordingly. So other than this, by default, the isolate mode is set to true. So that means it will carry out the MLST typing, the species identification, and etc. So including the AMR profiling. So if let's say all this looks okay, then we can proceed to the next parameters. But if let's say you have certain customizations that you would like to do for your samples, you can also set it over here. And then for advanced options, there are also options for us to manage the BACTA annotation using different database, and also the the parameters for the genome assembly. So all can be set over here. And most of the time, we just need to we do not need to set anything else if we are going to use the default parameters. And then miscellaneous options, this one we can set how many CPU cores or threads that we want to allocate for the analysis. If let's say we want it to be faster, we can increase the number of CPU threads over here, depending on the capacity of the computer. And then one last thing is the next flow configuration. So by default, every analysis run, the Epi2Me software will allocate or give a random name to the analysis. But if let's say we prefer to have a more specific name so that we can know what has been carried out in this analysis, then we can do the naming accordingly over here. So let's say this is a demo run on demo run on 4th June. Then we can name it accordingly to our based on our preferred needs. So once the setup is done, then we can click the launch workflow and then start the analysis. So the Epi2Me software will help to check whether all the required information or data is ready in the computer. So as you can see, the pipelines involve many steps, and then some of the steps might be using some database or data sets. So the Epi2Me will directly help to identify the required one and then do the download if required. So normally the first time running the analysis might take a longer time because it will take some time to download the required data sets in our computer. And then one is done or ready, the subsequent runs all should be faster compared to the first run. So once the analysis is being carried out, we can see the analysis page over here. So the status is running now. So under the details, we can see what other step is being carried out at the moment. So I think this assembly for 2 samples, based on my previous test, it's going to take roughly 15 to 20 minutes. So I will leave the process to continue run over here. While waiting, I can bring you to see the one that I have run yesterday. So this is basically the same run for the same data sets. So this one yesterday, it takes roughly 20 minutes by using the default parameters. So from this page, normally if you scroll down and then see all the green bars, then that means all the tasks or all the steps in the pipelines have been carried out successfully without any problems. So if you see any red bar, then that means some of the steps might fail. And normally we need to go to the log page to identify whether a certain step is stuck or it has been retried or retried and successfully redo the analysis. So once everything is done and then the status change to completed, normally we can take a look at the reports over here. This report summarizes the analysis results that has been carried out by the pipelines. So these reports include first the sample summary. So very quickly, it can tell the 2 samples that we put into the analysis, what are the species that they have identified based on the sequence. So the 2 samples that we have, bucket 01 are is basically the Mycobacterium tuberculosis, and then bucket 02 is Salmonella enterica. So it also helped to summarize what is the MLST or the sequence type of the 2 samples. And then the assembly, how many data, how many reads or how many sequence that we have in our data to do the assembly. And then after the assembly, how many contigs we obtain from the assembly. So from this data set, as you can see, bucket 02 is actually a very low throughput data set. It's actually a subset from one sequencing run. So you can see with around 7,000 of reads over here, it comes with, it managed to assemble into a quite fragmented assembly with more than 300 contigs over here. Well, if we have sufficient data, then the assembly will be kind of near complete or complete. So for bucket 01, it managed to assemble everything into 2 contigs. So for further information, scrolling down, then we can see actually bucket 01, the quality profile is actually quite low. So this is a normal profile for fast base calling. So as mentioned just now, this data set, the base call models has been manually put into the data set so that it can pass through the pipelines. So by default, the pipelines will only take the data that come from the high accuracy base calling and also the super accurate base calling. So for fast base calling, the pipeline might report an error. So that's why they manually fix the data set by changing the base call models over here. But this is a very, this is a fast base calling data set. So the quality overall is rather bad. So normally the high accuracy one or the super accurate one, we are seeing the quality now has been shifted all the way to almost 20. And then this is the rate length distribution for the data. And then the throughput is actually less than 200 MB. So normally this data sets with this size is not sufficient to do the assembly, especially if using other technologies. But this one is still managed to do the assembly. It's just that the outcome might not be, might be kind of suboptimal. So this is bucket 01. So if we want to look into bucket 02, we can also do the switching over here. So this is a more reasonable profiles, quality profiles for high accuracy base calling mode. And then the distribution, this is a very low number of kind of low throughput for samples. The distribution, the size, I think is relatively quite okay. So each of the rates that we obtain is roughly few KB. And then the throughput actually is very, very low, is just 24 MB. So yeah. This basically lists out the assembly summaries. So how many contigs that we obtain, and then for each of the contigs, how what is the length of it, and then how much coverage obtained for each of the contigs. So we can also do the sample switching over here in this report. And whether the pipelines identify any plasmid in the samples. So bucket 02, it identified 3 plasmid contigs out of the 300 plus total contigs over here. And bucket 01, obviously there's no plasmid identified. And then annotation, this is done by using the BACTA, so it identified the genes inside the genome. So we don't need to further identify another software to help us do the genome annotations after the assembly. So it can done all here in one pipelines. So this one have identified more than 5,000 genes inside bucket 02, the Salmonella samples, and then for bucket 01, it has identified more than 4,000 genes inside the samples. And the AMR profiling, so for bucket 02, identified 2 different AMR genes, and then the predicted to resist to these 2 drugs. And then another gene basically might help the bacteria to convert these particular drugs. So for bucket 01, yeah, this mycobacterium, I think likely is a MDR or XDR. So it kind of contains a lot of genes that helps with the antimicrobial functions. So yeah, all this is listed in this report. And yeah, this is also the reasons why I didn't really change the demo datasets for running these pipelines, because these two samples are actually some, I think, real samples where they identified some interesting findings from the sequencing run, even though the sequencing data throughput is very low, or one of them is of low quality. So the MLST typing, so the sample 2 is this particular sequence type, and another one is this particular sequence type is 215 based on the mycobacteria 2 profile. And then one of them is Salmonella samples, so the serotyping is carried out. So the samples for the Salmonella sample is actually this serotype, the Havana serotype, with the antigen profiles predicted to be like this. And then it also helped to profile the contact coverage, visualize the coverage plots over here. And at the bottom of the reports includes the software that is used together with the versions, and also some workflow parameters over here. So yeah. In addition to this bacterial genomes report, actually it also have 2 individual reports for each of the samples. So if let's say we have 96 samples, then it will also produce one summary report and then another isolate or sample reports for each of the 96 samples. So this one, it will have more details on each of the samples, including like the species identification, in addition to the mycobacteria, what is the second hit or second possible species based on the available sequence information. And then some additional informations for the samples are all here. So normally we want to also share the results with our collaborators or with our supervisors. So how do we share these reports? So our collaborators do not need to install the Epitome in order to view the reports. So these reports is available as HTML files. So we can get it over here in the output files list over here. So these files tabs listed out all the output file from the analysis workflow. So including the assembly in the FASTA format, and then the annotation in the GFF format, and etc. So that also include the reports in HTML. We can share the HTML reports with our collaborators, and then they will be able to open it in any web browser very easily to view the results. So each of these file or folders, we can click to open over here or to open in File Explorer accordingly. So my preferred way to look into the output files for certain analysis run will be directly from the options over here, and then open folder. So once it's opened up, then basically these are all the output files from the analysis run. So including the reports and all the results files. So from here we can further manage our output or analysis results accordingly. So yeah. So last part will be the logs. So this one, as mentioned, normally we don't need to come to this particular tab. This one is normally required when we want to check certain workflow details, and also if we run the analysis into errors. So if the status of the analysis is stopped with errors, then normally we will come to these logs to try to check what are the steps that goes wrong. So normally if let's say this thing is completed without any error, then this one can be skipped. And then for analysis, in case some of them run into problems like this, so the status will be stopped with error and then red color. So normally for cases like this, we can see which step actually runs into problems. And then from the logs over here, we can try to see what is the reasons that certain steps cannot be carried out accordingly. So here, normally it will help to troubleshoot what goes wrong. but normally this message, it might not be very kind of readily to be read by users. So one fast way to do the troubleshooting is you can copy the whole error message over here or the whole logs over here, and then paste it into any AI tools like ChatGPT or Gemini. It will help to troubleshoot for you. So what goes wrong, and then recommend or suggest what is the best way to rectify the problems. So once we identify the problem, so for example, like this case, this one is rather straightforward. So the error is caused by this particular step, and then the error is due to, I think, the models, the base call models used is not identified or not cannot be identified directly from the data. So in this case, normally we need to manually tell the pipelines what is the base call models that is used to produce the data. So this is some of the ways that we troubleshoot our analysis if, let's say, we run into problem. And in case some error is still very tricky, we are not able to tell what is it, and AI is not actually giving any helps in that. So the last resort could be over the same analysis run that start with error, and then there's a report issue button over here. We just click this report issue, and it will help or tells you how we can get the assistance from ONT, because the pipeline is developed by ONT. So that's why if there's any error, the ONT support team will be able to help to do the troubleshooting as well. So this to get the help from ONT support directly, we need to prepare some files for the team so that they can know what happens in the analysis. So we need to export certain files from the analysis run, and then these files should be sent over to the support at nanoporetech.com, and then inside the email describe the problems that you are having, and then attach the files that is exported over here. So yeah, so one is exported, then it should be in the folders over here. So it's a file in this particular format. We just need to attach it to the email and then send to the support at nanoporetech.com. So alternatively, in case you want to have a faster kind of somebody to help to look into the error logs and etc., you can also send these errors email to NextGene, so send to me. I will try to see what I can help to resolve the problem. So basically, this is how we carry out the analysis by using the Epitome, particularly for the bacterial genome or microbial genomes. So this particular workflow at the moment, it can help to do the bacterial genome assembly and all the further steps. So if we look into this map, somebody say this is our, it looks like our MRT map. So you can see the station by stations over here. And then the same pipelines can also be used for other microbes, including like fungus. And then if, let's say, you have more specific samples or applications, you can also check with NextGene or check with me on how to proceed with the data analysis accordingly. Yeah, I think that is all for the demonstration. So back to here. So just a quick summary of what we have gone through in the 1 past hour. So we have gone through briefly on the Oxford Nanopore technologies in terms of how the nanopore sequencing is carried out, and also what are the available devices or solutions available. And then we have also gone through briefly on the no-miss workflows on how to run the experiments or how to prepare the data before we do the analysis by using the Epitome program to get the whole genome of the bacteria and etc. So with this, I will end my sessions over here. So if you have any questions, please feel free to drop it to the chat box.
Yep, thank you, Jiaxun, for the insightful presentation. So now we will move into the Q&A session. You can unmute yourself or feel free to type your question in the chat box. Is there any question from the participants? Let's wait for another couple of seconds.
Seems like no question for this round.
It seems your presentation is very comprehensive and everyone is understanding. Yeah. So thank you, Jiaxin, again for the informative and comprehensive presentations today. And also a big thank you for everyone for attending this session. And we hope you find the information shared is valuable. Lastly, we would like to know what your thoughts for today's session. If you have a quick moment, please fill out the survey in the chat. And we will send the certificate once it is completed. Additionally, if you have any more questions after this webinar, you can drop us at email at sales@nextgene.my. Thank you once again, and we look forward to connect you in the next session. So, Muhammad Amiru asked for the presentation slides. Yes, we will share the recording and slides after it is completed. Yeah, thank you all.
Have a nice day.